When we think about the fight against cancer, most of us picture a battle against a tumor, right? A single growth in one spot. But what if I told you that the primary tumor isn't the real killer? The true challenge, the thing that has stumped scientists for decades, is stopping cancer from getting out. And that's exactly what we're going to break down today.
Let's just start with a number that'll really make you stop and think. 90%. That's how many cancer deaths aren't actually caused by the original tumor. They're caused by its spread to other parts of the body. This one process is, without a doubt, the single biggest enemy in cancer treatment.
And that process has a name you've probably heard: metastasis. You can think of it as cancer's great escape plan. When cancer cells break away from home, travel through the body, and set up new camps somewhere else, that's when the disease becomes so incredibly tough to beat, and way too often, fatal.
So you're probably wondering, well, why haven't we just made drugs to stop this? And the honest answer is, we've tried. But for years, these efforts just failed one after the other. It got so bad that the whole field was kind of put on the back burner. And the reason for all these failures? We basically misunderstood the problem. We were trying to stop the escape without ever really understanding the escape plan itself.
But that's where the strategy has completely flipped. The new thinking is, if you want to outsmart the escape plan, you have to take it apart first. You've got to deconstruct it piece by piece to find its weakest links.
So to do this, a whole new systems biology approach has come into play, creating this amazing set of interconnected platforms. Think of it like a complete toolkit designed to do three things: analyze the entire metastatic process, predict what it's going to do, and then rapidly test new ways to stop it. We're going to unpack this whole system.
Okay, so the very first step is, foundational. You have to be able to recreate that entire incredibly complex journey of a cancer cell, but do it on a lab bench. That's the whole point of this first platform. It's built to mimic every single part of that escape plan, just outside of a living body.
And that journey really has three main stages. First, you've got dissemination. That's when the cancer cells actually break away from home base. Second is travel. This is the really dangerous part of their journey, surviving in the bloodstream while the immune system is trying to hunt them down. And finally, there's colonization, where they have to land in a new organ and successfully set up a new tumor. This platform lets scientists watch and measure all three of those steps.
So, once you can actually measure this whole process in the lab, what do you do with all that data? Well, you use it to predict the future. This is the next piece of the puzzle, turning those lab results into potentially life-saving predictions for real patients.
And this is where biology gets a serious upgrade from artificial intelligence. A platform called MetScan takes all of that rich data gathered from patient tumors in the lab and feeds it into a machine learning algorithm. The big idea? To create a super powerful diagnostic tool that can look at someone's tumor and predict how likely it is to spread.
What's so cool is that we can actually get a peek inside the AI's brain. This kind of visual shows us exactly which biological factors the AI is paying the most attention to when it makes a prediction. It's like it's pointing a giant spotlight on the weakest links in cancer's escape plan, the steps that are the most critical for it to succeed.
So, does it work? You bet it does. In a blinded study where the AI had no prior information, the algorithm hit 92.3% accuracy. But here's the really important part. It correctly identified every single patient whose cancer went on to metastasize. We're talking 0 false negatives, which is absolutely critical for a tool like this.
Okay, so we can measure metastasis and we can predict it. What's the next major hurdle? Well, it's testing potential drugs. How do you find things that can actually block that escape plan and do it quickly and effectively?
See, this has always been a massive bottleneck in research. The traditional animal models for studying metastasis can take 4 to 6 months for just one experiment. They need a lot of animals, and honestly, they often have a success rate as low as 20%. It's just way too slow and inefficient for finding new drugs fast.
But check this out. A new mouse model called MetiVivo completely changes the game. The study time plummets from months down to just 6 weeks. You need way fewer animals, and most importantly, the reliability of the model just skyrockets, from a dismal 20% all the way up to nearly 100%. That is a monumental leap forward.
But what if you could go even faster, at least for the first round of screening? Well, believe it or not, you turn to the humble fruit fly. An even faster model uses them because you can get results in just 10 to 12 times days. And it turns out we actually share about 75% of our disease-causing genes with them, which makes them surprisingly useful. This lets researchers screen tons of potential drugs really fast to find the most promising ones.
All right, so this brings us to the big payoff. What happens when you put all these pieces together? The lab assays, the AI, the faster animal models. You get this incredibly powerful engine for discovering entirely new ways to fight metastasis. And that engine has led to a really mind-bending question. What if you could stop cancer from spreading without even touching the primary tumor? This idea is called uncoupling proliferation from metastasis, and it's kind of the holy grail of cancer therapy.
Here's what that actually looks like. With this new kind of drug, the original tumor might not shrink at all. But, and this is the whole point, the cancer's spread to the rest of the body is dramatically reduced. The drug isn't killing the tumor, it's just disabling its ability to escape and set up shop elsewhere. It's trapping it.
And this isn't just a theory. The system is already finding real, promising drugs in two different ways. First, there's a phenotypic approach, which is basically just trying things to see what works, and it's found existing drugs for other diseases that also happen to stop metastasis. And then there's a target-based approach, which is like finding the exact lock and then designing the perfect key. And that has uncovered brand new targets, leading to totally new drug candidates. Behind all of this incredible science, from AI algorithms to fruit flies, is a really simple, profoundly human goal. As the researchers themselves put it, the whole point is to offer patients a second chance by, quote, eliminating the term metastasis from the vocabulary of their primary tumor. And that leaves us with a pretty powerful thought to end on. By taking apart cancer's escape plan and hitting its weakest links, you have to wonder, could we be on the verge of a new era? An era where a cancer diagnosis is still serious, of course, but where its ability to spread is no longer the death sentence it's been for so long.